So this is a topic that ties the semester pretty nicely together. It brings us back around to what we covered with Punnett squares and predicting offspring, but also allows us to think more broadly about groups of organisms together. I realize you probably have had this in other classes. So if you took ecology or this gets covered in the biology 142 class, this might be familiar. I'm hoping that maybe if it didn't make sense the first time around, we can solidify some of that here. So this is the topic of population genetics. Population, of course, is a group of organisms of the same species that are able to interbreed. So this becomes a more interesting question if we look at scale of time and we look at evolution and things like that. We can explore bigger questions than just looking at the transmission of individual traits when we study population genetics. So a nice population of wildflowers there. So population, right? What is a population? Group of the same species individuals. And in population genetics, we're looking at a group of alleles inside of the individuals of that population. So we're kind of thinking about the alleles outside of individuals. We're thinking about alleles kind of floating around and asking the question of whether those alleles are being traded around equally in a large population, or maybe if there's some kind of factor that's causing them to not get mixed up equally. So for example, this is a study that dates back to Wisconsin, and an interesting revelation here with some salamander populations. So one of the ways that you can learn from studying population genetics is to establish that evolution is happening. So this is maybe one of the quickest ways, or one of the more intimate ways to get familiar with evolution actually happening in populations. And we may not see it initially as drastic changes in organisms, but might see the beginnings of these changes. And so population genetic tools can be some of the first evidence you get to see big scale changes. So this is an interesting study where they found out that these salamanders were evolving to remain in the water. So typically salamanders go through a metamorphosis, as do many amphibians, and they crawl out of land or out of water and live on land. So they're eggs in the water to begin with, and then as adults they live on land. And this population was living in the water for its entire life, which is a little bit weird, but it kind of fits if you look at the nature of this water body. This is a water body that was, because of human activity, kept warm year round. So these are salamanders that did not freeze during the winter, and they actually evolved to take advantage of that and stay alive and stay active in the water. So we're going to look at this idea of alleles moving around in a population, and we're going to get back around to one of these more mathematical evaluations to ask the question of whether things are, whether reality is fitting with our expectations or not. And so I just want to establish that we're looking at null expectations. And if you remember, we used this term null hypothesis or null predictions. We used that before when we talked about the chi-squared test we were doing with our fruit flies. So we had this kind of background expectation or possibility. Let's say we're entertaining this possibility that things are behaving kind of normal, as expected, no funny stuff, normal predictions. And in population genetics, and what we'll get to with the Hardy-Weinberg equation, the null expectation is that everybody has a chance to mate with everybody else, that the mate choice is essentially random, that the population is large. And therefore, in this situation, these alleles have the ability, not really ability, but the alleles will be found basically randomly through the population relative to their abundance. So kind of on the topic of random mating, I like to throw a little bit of trivia in once in a while. This is an artist's project, a photographer's project, where they were set out to document people who are of mixed ethnic heritage. So it's kind of highlighting in this article how the future of people, particularly in a country like the United States, where we have people from lots of different ethnic backgrounds, the future is really for all of these people to start, sort of break down their heritage barriers, and for more individuals to be born with mixed ethnic backgrounds. And so this photographer chose people who are a little more noteworthy as being born from parents or recent ancestors of a little more broadly dispersed ancestral heritage, possibly a little window into the future. So if you took all the individuals who live in North America and allowed everyone to have random mating with everyone else, this would be closer to what our future would look like. An interesting sociological question, really, when you think about what the future population of the United States might look like. So in more of a natural setting, if we had a population of organisms that have been in a place for a while, and we expect that they've been mating randomly over that time, we expect more of a homogeneous mix of alleles in the population. So kind of a contrast to what I'm showing here with these images. These images are the result of people whose ancestors came from very different parts of the world, where they had been isolated from other people. So the ancestral populations in Africa, before people were sailing across oceans and now flying across oceans, before that time, there was no chance for a person living in Africa to meet anybody who lives in, say, northern North America or Australia, for example. And now we have this possibility for people from all different backgrounds to have babies together and make happy lives together. And so this is really kind of counter to our null expectation, which is something we have more for natural populations. So sometimes when we break the expectations we have for our null hypothesis, then we find something interesting. So in a natural population, the phenotypes stay roughly the same, because those individuals are a population of interbreeding individuals. And as I said a few times, the chance that any one person or individual will mate with another individual is essentially random. So no mate choice or selection or anything like that. So as I've said before, we're thinking about our alleles kind of outside of the bodies of the individuals that have them. So if any one person can mate with any other person, then we're really just a collection of genetic material that is a sample of what's already out there in the population. So this idea of sampling, this is why I chose the lottery ball picker, bingo ball picker. When a person gets their genotype, it's basically a random sample of what's out there. So it's a sample of alleles across all the different genes that have different alleles in the population. So a person may be homozygous for one allele, heterozygous, homozygous for the other allele, and then that same pattern really across all genes. So this is where we get this term gene pool from. You probably heard that before. So we draw from the gene pool. And I kind of like that idea, right? So if you sort of took all the alleles out of the people, put them all together and randomly picked a pair, each person gets 2 alleles, that would be one of those possible genotypes. So we're going to keep things kind of simple when we look at this topic. We're going to look at one gene and 2 alleles that belong to that gene. And don't worry, it still gets pretty complex. So let's say we have alleles, blue alleles and brown alleles. How many ways can you mix up alleles big B and little b? How many possible genotypes? Well, not too many. So you can have homozygous for big B, heterozygous big B, little b, or homozygous for little b. And those different genotypes, you can calculate the frequencies of genotypes in the population. So again, we're looking at multiple individuals, and now we can take their genotypes and look at them more from the perspective of just their alleles in the population. So if we have these types of individuals, simplified example where we have the big B allele causes a person to have brown eyes, whether they have one copy or two copies of big B, they still get brown eyes. So I guess important to remember while we're doing this that we are looking at genotypes and not phenotypes. And we're also, I guess, generally looking at situations where there's no adaptive advantage, maybe even not a phenotypic difference between these different genotypes. Just sort of something in the population that we can use to track where people are coming from, or individuals are moving from, or staying in place. So we can take these numbers of individuals, and I've made them conveniently add up to 10,000 individuals, and we can figure out how many, what proportion of our alleles, if we take all the alleles, how many of those are big B and how many of those are little b. So let's start with our frequencies of genotypes. So frequency here, frequency is the decimal indicating proportion that are this one kind. So 4,800 out of 10,000 is 48%, or 0.48. 4,400 out of 10,000 is 0.44, or 44%, and 800 would be 0.08, or 8%. And if we took those numbers and added them up, they should be equal to 100. So again, in our simplified example, we're going to consider that we have accounted for all possible genotypes using just these two alleles. So that's not too hard to calculate, especially if you have a total of 10,000 individuals in your population. What about the chance of inheriting alleles? Well, the chance of inheriting an allele, right, big B or little b, the chance of getting an allele, if we're just putting them all into the gene pool and drawing them, then your chance of drawing a little b is proportional to the number of little b's in the population. So if there are 70% of your alleles are little b, then 7 out of 10 times you should be drawing a little b, right? So let's see if we can take these genotypes and turn them into allele frequencies. So let's start over here with the little b, little b. So there are 4,800 individuals that have the little b genotype. And each of those individuals, we can think of them as having two alleles, and both of those alleles are little b. And that's different from these individuals that also have a little b, but only one of their alleles is little b. So it's basically 2 times the number of homozygous little b individuals plus 1 times the number of heterozygous individuals. And this is the formula you can use to calculate those. So like I just said there, 2 times the number of, let's see, we'll do the bottom one, 2 times the number of little b individuals plus the number of big B, little b individuals. As I said, that's the number of alleles in the population that are little b. And we want to divide, to get the frequency, the number of total alleles, which is all the people in the population, and 2 alleles each. So 2 times that total. If we do this right, these two values should add up to 1. The frequency of the little b allele here, we're calculating and calling that Q. The frequency of the big B allele, can calculate that in a similar way, and that gets called P. P and Q are commonly used in this equation, and this is actually a variation on the equation we saw earlier in the semester. We saw the binomial expansion. So the P's and Q's here are a binomial expansion where we have two of these together. The first one is P or Q. The second one is P or Q, and the combination of those gets us the binomial expansion that we will see in a little bit here. Another way to calculate this, if you've already got the frequency of genotypes, then you can use your genotype frequencies to convert those right into allele frequencies. So if you know the frequency of homozygous big B individuals and the frequency of heterozygous individuals, then the frequency of big B in the population, this allele frequency, simply this number plus half of this number. And this calculation takes advantage of the fact that the genotype frequencies add up to 1, and these should add up to 1. So we're adding all these things together. We're taking half of the heterozygous individuals and putting them in the big B category, and half of the heterozygous individuals and putting them in the little b category. All right, so let's see if we can calculate one of these. Let's say we have a population with big B is 20% of the alleles, little b is 80% of the alleles, or 0.8. What are the chances that those will combine and give us an offspring with the genotype little b, an offspring with the genotype big B, little b? So for that, we're actually taking a variation on the old Punnett Square. And it's the Punnett Square we derived earlier to predict the outcome of one mating, where we knew the genotype of one parent and the genotype of the other parent. And we're going to use the same Punnett Square, except this time we're going to turn these into basically odds that are unequal. So the odds that the female parent donates a big B allele, that's not equal to the odds of donating a little b. So these are proportional to the abundance of these alleles in the population. So 20% of the alleles are big B. There's a 20% chance, 2 out of 10 chance, that the female parent will give a big B to her offspring. There's a greater chance, a 0.8%, an 80% chance that the female parent will donate a little b to her offspring. So to combine these together, any one of these two combinations, if we want to ask what are the chances of the offspring being big B, that's the chance that mom gives a big B and dad gives a big B. So this was a 0.2 chance from mom, a 0.2 chance from dad. We need both of these things to happen to get this genotype, so it becomes P times P, or P squared, which here is 0.04. Same thing for the B's, 0.8 and 0.8. Both of those have to happen together. 0.8 times 0.8 is 0.64. The other one, the last category, is a little bit more tricky because there are two ways to get this. So to become big B, little b heterozygous, you can get either a big B from dad and a little b from mom, or a big B from mom and a little b from dad. So these two squares show the same value, and we need to add them together to get the total number of ways you can get big B, little b outcome. So it's actually 0.16 plus 0.16. Again, all of these numbers in these 4 squares should add up to 1. All right, so what we did there, we did it, but we didn't say it just yet. These numbers become the hopefully familiar figures in the Hardy-Weinberg equation. So a population that's in equilibrium of random mating, large population size, then we have this expectation that we'll have this many, the frequency of homozygous individuals for one allele, heterozygous individuals, and homozygous individuals for the other allele. P squared, 2pq, and q squared. So I mentioned equilibrium. What is equilibrium? Well, I have a little diagram, or a photograph I chose to show us as rock balancing. Balancing is really the nature of equilibrium. That's what that term is based on. So equilibrium is balance, and when we talk about genetic balance in population, this means that future generations will have the same allele frequencies as the present generation. Another way to say that, as I have it written here, the allele frequencies will stay the same over generations, the genotype frequencies will stay the same over generations. So again, if we have a population where interbreeding has happened randomly over generations, then this should be the case, that we can just see these alleles getting equally traded among all reproductive individuals. It also relies on some expectations. So you might have seen these before. This equilibrium anticipates that there are large populations, that everyone's allowed to mate randomly, and that there are no outside forces that are influencing survival or reproduction, or there's no combinations of individuals between populations and so on. And so if any of these things happen to a substantial degree, if we break any of these expectations, so population that's a little too small, individuals that are not randomly mating with each other, movement of individuals between populations, if those happen to a substantial amount, then we'll be breaking our expectations for Hardy-Weinberg equilibrium. And we should be able to calculate these as a deviation from what we expected. All right, so here's an example. Let's say we had these number of individuals in a population that now adds up to 1,000. We know the genotypes, we know the number of individuals with each genotype, and we could turn those into a frequency. That's pretty easy in a population of 1,000. So 100 out of 1,000 is 0.1 here, and so on. We can go from either of these to calculate our allele frequencies. So the frequency of the big D allele is our P. That is, you could do it here. So 0.1 plus half of 0.2 is 0.1 plus 0.1 gives us 0.2. Could calculate that other ways as well. And then once we have our allele frequencies, we can go to a predicted expectation. So the direction is really, these arrows are important. You've got to go one step and then the next step to be able to calculate these values correctly. So population, the expected frequency of big D individuals is P squared. 0.2 times 0.2 is 0.04, and so on. And if you want to take those expected values for frequencies and turn them into expected numbers, then you take, like we did previously with the chi-squared test and the fruit flies, take your frequency, multiply it by the total number of individuals. 0.04 times 100 gets us 40 expected individuals of the big D genotype, 320, 640. Those add up now to 1,000, or the total population size. And then it's these things, these values, the observed actual numbers and the expected actual numbers, those are what we compare in this chi-squared test. So remember, we got to calculate chi-squared using actual numbers, and that gives us the benefit of, we get a better statistics, we get kind of a more reliable statistic if you count more individuals. So a population of 1,000 is better than counting a population of 100. Counting 10,000 individuals would do even better for this calculation, a little more, like I said, statistically reliable. So that's our chi-squared. Chi-squared involved taking expected and actual numbers, and also our degrees of freedom. And degrees of freedom, if you remember, was our number of categories minus 1. So we've got 3 genotype categories, so 2 degrees of freedom moving forward. And I've got it all calculated out here for you. So observed, expected, just kind of eyeballing this, we're not too far off. So 100 versus 40, that's pretty far off, but 700 versus 640, maybe less far off. Unfortunately, this being off by a lot in the small category, that means that we take this 60, square it, and divide by 40, that gets us a pretty big number. So altogether, when this is all calculated, the final total chi-squared statistic is 140.6, and no matter how many degrees of freedom you're looking at, we're way off the charts. So this is an extremely small p-value. What do we do with a small p-value? Well, that brings us all the way back around to our null expectation. So the null hypothesis was that the population was interbreeding everybody had a chance of interbreeding, no migration, no selection, random mutation, or random matings, and so on. So something about this population might be deviating from what was expected, or could be, as we saw in the fruit fly experiment, sometimes we come around to explaining this based on not having enough data or something like that. So something has gone wrong that prevented us from getting what we thought we should get for the outcome. Maybe it's biologically meaningful. Maybe we messed up. If we trust our data, then we could say there's probably a reason, some kind of biological reason why. For example, there's a lot more, it looks like, of the big D individuals than we expected. Maybe there's a reason for that. So the chi-squared test of the Hardy-Weinberg expectations kind of gives you a starting point for investigating something else, maybe coming up with a nice explanation for why you got the deviation you did. All right, so just kind of to wrap it up, this would be the order of calculating things, just like I did in the diagram with the arrows. Do one and then the other. Don't skip any steps. You can get the wrong numbers if you calculate in different directions than what we did. So figure out your genotype frequencies, figure out your allele frequencies, turn those into expected genotype frequencies, and then compare your observed, not only the frequencies, but observed and expected numbers using that chi-squared test. And then last step, if it's a difference that's significant, try to explain it. Always a good ending for biology, right?